If muscle glycogen is used for anaerobic glycolysis, then how many ATPs are obtained or formed? Okay, so what is the main clue here is this, that end product of muscle glycogen is glucose 6-phosphate. Okay, what is the end product of liver glycogen? When liver glycogen is broken down, then we get free glucose in the end, and this free glucose can escape from the liver cell into blood, and that's why liver is responsible for maintaining blood glucose, right? So in liver, glucose 6-phosphate gets converted to glucose in the end. This step is done by enzyme glucose 6-phosphatase. So this enzyme is present in liver but absent in muscles. So in muscles, when glycogen is broken down, in the end we get glucose 6-phosphate because the enzyme for the conversion of glucose 6-phosphate to glucose is absent. The enzyme is glucose 6-phosphatase, right? So this glucose 6-phosphate will enter into glycolysis in muscle cell to give energy, right? Now, when glucose 6-phosphate is directly entering into glycolysis, then there is no need of hexokinase to phosphorylate glucose. Okay, so there is no need to use this ATP at hexokinase step. Right? Now they are talking of anaerobic glycolysis. In anaerobic glycolysis, our energetics is 4 - 2. Okay, what is this 4? Substrate level phosphorylation. What is this 2? 2 means one ATP used at hexokinase, other ATP used at phosphofructokinase 1. Okay, but now hexokinase is not used, so with this will be 4 - 1. 4 - 1 will be 3 ATP. So our answer to this question is 3 ATP.